Phonetics lab, I linked 2 things because you have to flip coins. The first, this bottom one right here is like the one that you've used. Sorry, I'm not sharing. There we go. This bottom one right here is the one you've used before when we did the making babies activity and we had to figure out which chromosome was being passed on. But we also have to flip 2 coins in one situation. So there's the 2 coin flipper. Okay, so let's open our lab. It's a blocked website. I'll find another one for us. Okay, so we're gonna start here and then I'll find you a second. There's like 2 websites, so I'll find you another one. All right, so how well, the guiding question is, how well does a Punnett square predict an actual ratio of offspring? So in this lab, we will make predictions using Punnett squares and pennies or virtual pennies to simulate the crosses. Then we will compare our actual ratios with our predicted ratios. The trait we're looking at is the gene that codes for a short toe in humans. An uppercase T represents the dominant allele, which would be a short toe, and the lowercase t is the recessive allele, which is a long toe. The following genotypes are possible. So if we got 2 uppercase T's, what would that look like? Short or long? Yeah, short is dominant. What about the second one? If we get heterozygous, one of each? Yes. And in person, it is still a short toe because the dominant trait only needs to show up once in order for it to show up in the individual. And then 2 lowercase, recessive. What kind of trait do they get? Yes, remote and in person are correct. Okay, so then using a Punnett square, we're going to predict the ratio of offspring in a cross where both parents are heterozygous. So I already set it up for you with the uppercase, lowercase, one of each. And now we just have to fill it out. So you guys can do this with me or you can go ahead. But I'm just going to fill in our square, how we've been practicing. I always start with the top. That's just me, though. You can do it however you want to. I bring my uppercase T down, and then I'm going to bring this lowercase t down to each row. And I bring this uppercase T across the top row. There's a space there. There we go. And this lowercase t across the bottom row. And I just always make sure you save it, otherwise it goes away. All right, then we're doing our percentages again, like how many out of 4. So how many squares do we get out of 4 that have 2 uppercase T's in one box? 1 out of 4, and what percent? Yes, so we got 25. Whoa, what just happened? Oh gosh, hold on. There we go. Whew. 25%. And then how many have 1 of each? How many squares, or what percent? Either way. Remote, did I lose you? Where are you at? And then the last square. 1 square has 2 lowercase t's together, so what percent is that? 25%. Okay, so then we're just looking at the phenotypes. Yeah, there you go, guys. So our phenotypes, if we have, remember, short toe is dominant, so it's an uppercase T. We have to add the 2 together. So 50 plus 25 is how much percentage would have a short toe. You're right, I'm just waiting for it. Yes, okay. Everybody got 75, and then if you just take 75 minus 100, or you look at the recessive percent, it's 25 for a long toe. Okay, so I highlighted these. If you notice, they're kind of like orange because later on down here, you're gonna want to fill these in. So the orange matches with the orange. So I made the colors match with each other so that you knew what part was which part. Okay, I'm gonna set you up for what you guys are doing now. So now we are going to determine the actual ratios by using coins to represent the crosses. You are going to use 2 coins. On one side of the coin is heads, which represents the short toe, uppercase T. On the other side of the coin is tails, which represents a long toe, lowercase t. The coin represents a parent, each coin represents a parent that has the genotype heterozygous. So the second coin, obviously, like I said, is the other parent. So you have 1 coin representing mom, 1 coin representing dad. I just said that. Okay. When the coins are flipped, you are determining what sperm or what egg trait is being donated or given to the match. And when you put the 2 flipped coins together, you are simulating fertilization. So here we're just looking for, just to write this down so that you can have it here. I said heads is the short toe. So it's just reminding us when you're flipping coins. And tails is long. All right, so in order to determine the actual ratios, you're gonna flip a coin 50 times, and I picked that one because you could just set it up, but I'll find you another one. And you're just gonna record it on the paper. So if you want, as you're flipping, you can just write down little like tally marks, like a 1 or whatever. However you wanna record it, you can write it on a sheet of paper, whatever works best for you. I don't really care if the tally mark column is filled out. It's just how you can record or like track how many there are. Like I said, you may want to just tally it on a piece of paper, which I have scratch paper if you guys want it, if you don't have anything to write on. And then once you get it out of 50, you just add them up or put your total number here. And then because it's only 50 and not on 100, you're going to multiply by 2 to get it out of 100%. So I'm not making you flip it 100 times, basically what I'm saying. So you might only get like 10 here, 35 here, and then I don't even know the rest. 55 there, and then you just multiply it by 2. Well, no, sorry. 55 is more than 50. I can't math in my head. Website, if it does work for people that aren't here in the school, just do unflip like the side landing, because otherwise it can end up on its side, and that's dumb. So just unclick that if this one works for you. Okay, so I just, for part 3, like I said, I set up your Punnett square. So you're going to end up bringing this capital T down to both of these, this lowercase t down to both of those on the right side, and then lowercase t's on the top row and lowercase t's on the bottom row. So fill that out. You should have gotten something like that. If you're still working, that's fine. You can check your work. And then for our percentages, remember it's out of 100. So how many boxes out of 4 have 2 uppercase T's together? 0. There's no possibility of getting a homozygous dominant. So it's just 0%, 0 out of 4. And then the only 2 left is there is heterozygous in 2 boxes and homozygous recessive in 2 boxes. So 50-50 split. And that would actually end up being the same for our phenotypic ratios, because the short toe, remember, is the dominant trait, which is, if it's heterozygous, you end up showing the dominant trait. So it's 50% because you don't need to add anything from the top because it's 0. And then 50% again for the recessive trait, because 50% of the box had 2 lowercase t's together in 1 box. So then you're pretty much going to be doing the same thing with flipping your coin, except now you only need one coin. So if we go to the classroom and I gave you guys, it should be this one. I just typed in Google, like coin flip. It's just like what we did with the Making Babies lab. It's the same thing. So there it flipped, it's heads on the, oh, I'm not sure. Sorry, guys. It just flipped and it flipped onto heads for my remote group. So it's going to be heads and tails was my first one. Because if we look at the parents, let me just go back and tell you the reason why. So we look at the parents, say this was mom, she's always going to give a recessive or the lowercase t. So no matter what, if you were to flip a coin, her heads is going to be a lowercase t, her tails is going to be a lowercase t. So it doesn't matter. All that matters is what flip you get. So for the dad, let's say. So that's why we're only flipping one coin, because only one of the parents can give off another trait. So this one, technically, you should get 0. You should be able to fill this in right away. There's no possible way for you to get 2 uppercase T's. So you can literally just fill in 0 because we figured that out. Oh, sorry, Irma, I keep forgetting to fill it in. But I was telling you that the mom will say it always gives those lowercase t. So it says it right here. We only need to flip one coin because the other coin will always have the lowercase t to contribute. So we can go again. So I'll flip it again. I get tails. So I go over here. Now I got 2 tails. Again, you guys can use this data too if you want, if you didn't already do it. I got heads. So I get another heads. So now you're just flipping one coin and doing the same thing. There are questions, but you guys don't need to turn this in until tomorrow. So as long as you get the flips done today, that's plenty of time. At 10:30, I'm going to start your exit ticket where I'm going to show you the character strong video. So you have, what, 20 minutes to work right now until I give you the character strong video? So like I said, as long as you get your 50 flips done, you're good for today, because tomorrow we'll be finishing up this lab. If you get stuck on something or you need me to scroll back up, just let me know so that I can show you what you need or share with you.